Hi everyone, it's Dr. LeBlanc with Unit 15B on Viral Genetics, the Complementation Test. Our learning goals are to understand how the complementation test using T4 viruses was used to map different genes, INTRAGENIC mapping, and to understand how the complementation test was used to make fine structure maps of multiple mutations within a single gene, or intragenic mapping. There can be genetic differences in T4 plaque morphology. Here we have an example of two different morphologies of T4 viruses. This is what a wild-type plaque looks like, and this is a mutation causing something called rapid lysis bacteriophage. These plaques form very quickly and they're very large, but they often don't have very many particles in them. A simple question: are the mutations that cause similar plaque morphology in different T4 strains in the same gene, or are they in different genes? So we have several strains of T4. They have similar phenotypes. Are they caused by mutations in the same gene, or are they caused by mutations in different genes in the two different strains? In order to answer that question, we need to perform the complementation test. You start out with two mutant T4 strains, and they infect the same cell. Neither strain can produce plaques on its own, in this case, when they're infecting this particular strain of E. coli. If no wild-type plaques appear, it indicates that the mutations are in the same gene. That's non-complementation. However, if you get wild-type plaques, that probably means that the two mutations are in different genes. That is the case of complementation. Let's look at an example with some diagrams of genes. So we have two strains, and they both have the same phenotype, an R2 strain and another R2 strain. They both give similar phenotypic results. If the mutation is in the A gene in the R strain number 1, and also in the A gene in strain number 2, when you use these two bacteriophages to infect the same E. coli cell, you get no plaques, because neither one of these can supply a good copy of the protein that's produced from gene A. This is a mutant copy, and this is a mutant copy. No good gene A proteins, and they're necessary for plaque formation, so you get no plaques. No plaques, therefore the mutations are in the same gene. That is non-complementation. Okay. What if instead the strains have mutations in different genes? Here's a strain number 3. It has a mutation in gene A, and strain number 4 has a mutation in gene B. When you use both of these bacteriophages to infect the same cell, you get some plaques. That's because strain 3 can provide a good gene B protein, and strain 4 can supply a good gene A protein. And so you have all the proteins that you need in the same cell to make a plaque. That's called complementation, and therefore the phage mutations now we know are in different genes. What about mutations that are in the same gene? Can we map the distance between those two mutations within a single gene? Well, Seymour Benzer in the 1940s designed an experiment to do just that. He studied bacteriophage T4 mutants, designed the R2, the ones that we were just talking about. The R stands for rapid lysis. In intragenic mapping, the goal is to determine the distance between two different genes. Okay? Separate genes the distance on a genome between the two different genes. In intragenic mapping, the goal is to determine the distance between two mutations within the same gene. Clearly, the distance is going to be much smaller for mutations within the same gene than it is when you're mapping two separate genes in the genome. How did he do that? He used recombination tests to create fine structure maps of the genes within the R2 locus. Now, fine structure maps are something that we now know, we can now appreciate as mapping mutations to individual nucleotides within the gene. At the time Seymour Benzer did these experiments, we did not know that DNA was the genetic material, and we did not know what the structure of the helix was. Well, we did know that DNA was the genetic material, but we didn't know what the structure of the DNA was, when we had no really good understanding that a gene, the physical presence of a gene, was a sequence of nucleotides within a chromosome. Okay, so Seymour Benzer did all these experiments without knowing that. Intragenic mapping, again, is we're trying to find out the distance between genes. Intragenic mapping means we want to know the distance between mutations within the same gene that we know are different. Okay, so we have to set up this experiment. Seymour Benzer used R2 mutants that behaved differently in 3 different strains of E. coli. All of these mutants had the same characteristics. In E. coli B, the R2 phage produced unusually large plaques that had poor yields of bacteriophage. In E. coli strain K12S, the R2 phage produced normal plaques that gave good yields of phage. That's important because he needs to have a source of these phage. In E. coli K12, which has phage lambda DNA integrated into its chromosome, the R2 phage produced no plaques at all. So we have 3 different strains of E. coli that these R2 mutants either produce large plaques, normal plaques, or no plaques at all. As expected, the wild-type phage could infect all 3 strains of E. coli. Okay, so he wanted to test to see how frequently a wild-type virus could be obtained when 2 mutants were used to co-infect the same cell. He relied on recombination frequencies between the 2 viruses. Intragenic recombination is a rare event compared to intergenic recombination. The more frequently wild-type virus was obtained, the farther apart the mutations were, because you had more of a chance to get a recombination event when 2 mutations are far apart. The less frequently a wild-type virus was obtained, the closer the mutations in the 2 strains were. Okay, this is a conceptual diagram of what we think is going on. We have gene A with an R2 mutation here, another strain with an R2 mutation towards the end of the gene. This one is towards the beginning of the gene. Okay, when you co-infect these 2 together, you allow for the rare crossover event within a gene in the regions between these 2 mutations. Okay? And the result of this recombination is a double mutant and a wild type. This wild type, of course, will produce plaques in the strain that this one cannot produce plaques in. So these R2 mutations here and here cannot form plaques on the E. coli K12 with the lambda integrated into it. Okay? But this one can. Okay, let's do this experiment. You have 2 different non-complementing R2 phage. Okay, so we know that the mutations are in the same gene because they're non-complementing. Okay, you co-infect these into some E. coli B bacteria. The B strain is the one that we know that these phage can infect. Okay, so you isolate the new population of phage. It will contain mainly non-recombinant phage, but it will occasionally contain intragenic within the same gene recombinants of wild type and double mutant phage. Okay. Okay, so here's our wild type phage, and here's our double mutant phage. They don't look any different. The double mutant don't look any different than the single mutant one because they don't they all of these don't have functional A gene. But this one, however, will be able to make plaques. Okay, at this point, then you're going to take all these bacteriophage and then you're going to split them, the preparation, into two different groups, and you're going to infect two different strains of bacteria. Up here, we infected the E. coli B strain, which would allow all of these phage to grow. Okay, now we're only going to use the strains that will allow just the wild type to grow, as well as all of these phage, so that we can get a titer. Okay, so here we have the E. coli B strain. Okay, every single phage particle here will be able to infect it, and we're going to see how many plaques we get. Okay, we use a very small dilution or a small concentration of the bacteriophage because all these particles are going to be capable of infecting. We're going to use a whole lot more bacteria, phage particles, excuse me, to infect the E. coli K12 strain lambda. The only phage that can infect this bacteria strain are the wild type phage. Okay, and we'll count the number of wild type phage there. So we have 66 plaques when we have a very high dilution, and we have 11 plaques in this strain of bacteria when we have a much lower dilution. We can use all these numbers to calculate the recombination frequency between those two mutations within the A gene. We can calculate the total number of plaques by a multiplicity of infection. So the total plaques times the dilution factor of 10 to the 8th is 66 PFUs, plaque forming units, times 10 to the 8th equals 6.6 times 10 to the 9th. That's the total number of plaques we have in our test tube. How many recombinant plaques do we have? Well, the recombinant plaques are the ones that could infect the E. coli K12 lambda bacteria, because those bacteria can only produce plaques if you have wild type bacteriophage infecting them. So we had 11 PFUs times 10 to the 6th. Okay, we had a different dilution here. And that yields a number of 1.1 times 10 to the 7 plaques. So what was the recombination frequency? We take this and divide it by this, and we get a frequency. We have 1.1 times 10 to the 7th divided by 6.6 times 10 to the 9th. Okay, that gives a number of 0.15 times 10 to the minus 2, or 1.5 times 10 to the 3rd, which is this. Or if we multiply it by 100, it's 0.15%, a very low number. As we would expect for two mutations within the same gene. So in summary, intergenic mapping defines the distance between separate genes in a genome. Intragenic, or fine structure mapping, defines the locations of different mutations within the same gene. Mutations in two different genes can often give the same phenotype. Complementation occurs when co-infection of two viral strains with the same mutant phenotype yields plaques with a wild-type appearance. Non-complementation occurs when two different viral strains have mutations within the same gene. Co-infection of the two strains does not yield the wild-type phenotype. Bind structure, or intragenic mapping, involves co-infection of two mutant strains with different mutations in the same gene, and the selection of recombinant phage. The number of revertants, compared to the total number of phage particles, gives the recombination frequency between the two mutations within the same gene. This number is correlated to the physical distance between the two mutations.